All right, let's get right into it. We're going to tackle one of the biggest questions in all of biology. How in the world are traits passed down from one generation to the next? Today, we're unpacking the core principles of inheritance.
I mean, it sounds almost silly, right? An elephant has a baby elephant. Of course it does. But the why gets to the very heart of genetics. This simple fact that like produces like is the whole basis of heredity. But then, why aren't you a perfect copy of your parents? Why are siblings so different? That's where the real mystery begins.
You know, for thousands and thousands of years, this was just a massive puzzle. People knew something was being passed on from parent to child, but the actual how was a total black box. And that question is the central mission of genetics, the study of how all this stuff gets passed on.
So to even start solving this puzzle, we've got to get two key ideas straight. First, you have inheritance. That's the actual process of traits being handed down. And second, you have variation. That's the reason why offspring are never just carbon copies of their parents. These two concepts, they're the bedrock of our whole story.
And what's so wild is that our ancestors were masters at using these rules even though they had no idea what the rules actually were. They totally took advantage of natural variation. Think about selective breeding, creating amazing animals like the Sahiwal cows in Punjab, which are fantastic milk producers. They knew what worked, but the science behind it, a complete mystery.
That is, until the 1800s. And this is where our hero enters the story, an Augustinian friar named Gregor Mendel. He's the detective who finally started to crack the code.
For 7 solid years, Mendel was just meticulously, painstakingly running experiments on everyday garden peas. He published all his findings in 1865, but, and this is incredible, his revolutionary work was pretty much ignored for 35 years. It wasn't until 1900 that a few other scientists stumbled across it and realized what a bombshell it was.
Mendel's real genius was in his method. It was so elegant and so clean. So here's what he did. First, he started with what are called true breeding plants. You know, a tall plant that only ever makes more tall plants. Second, he crossed it with a true breeding dwarf plant. Then, he just sat back and watched that first generation, the F1. And finally, he let those plants pollinate themselves to create a second generation. This simple 4-step process, well, it was the key to everything.
And here's the first big shocker he found. When he crossed that pure tall plant with the pure dwarf plant, 100% of their kids were tall. Every single one. It was like the dwarf trait just poof, vanished into thin air. So the big question was, where did it go?
And this is the big reveal. When Mendel let those tall F1 plants reproduce, the dwarf trait suddenly came back. And it wasn't just random. No way. It showed up in this amazingly precise ratio. For every 3 tall plants, there was exactly 1 dwarf plant. A perfect 3-to-1 ratio. That's not a coincidence. That's a pattern.
Now, to explain this, Mendel came up with the idea of factors being passed down. Today, we call them genes. And he figured that for each trait, like height, you get different versions of that gene, what we now call alleles. So think of the gene as the recipe for height, and the alleles are the different versions, like the tall recipe versus the dwarf recipe.
And this is a super important distinction to make. You've got the genotype, that's the actual genetic code, the combination of alleles an organism has. Maybe it has 2 tall alleles, or 1 of each, or 2 dwarf alleles. Then you have the phenotype. That's the physical thing you can actually see with your eyes. Is the plant actually tall or dwarf?
This little grid, the Punnett square, is such a brilliant way to visualize how it all works. It basically shows you the odds. When you cross 2 of those F1 plants, their reproductive cells can combine in 4 possible ways. You end up with 1 TT genotype, 2 TT genotypes, and 1 TT. And that underlying 1 to 2 to 1 genetic ratio is exactly what produces the 3-to-1 physical ratio we see. It all lines up.
So from these incredibly simple experiments with peas, Mendel figured out 2 absolutely profound laws. The law of dominance explains why those F1 plants were all tall, the tall allele simply overpowers or masks the dwarf one, and the law of segregation explains that these 2 alleles split up when reproductive cells are made, which is why that hidden dwarf trait could pop back up in the next generation.
But, you know, just when scientists thought they have the rule book figured out, they found out that nature, well, it loves to throw a curveball. Mendel's laws are the perfect foundation, but sometimes the rules bend in some really cool ways.
Okay, so what Mendel saw was complete dominance, right? The TT plant was just as tall as the TT plant, no difference. But then you look at something like a snapdragon flower. You cross a red one with a white one, and you don't get red, you get pink. It's a blend. That's called incomplete dominance. The result is a mix, an in-between of the two parents.
Now don't mix that up with codominance. It sounds similar, but it's totally different. It's not a blend, it's more like both traits show up at the party. The absolute best example is human ABO blood type. If you get an A allele from one parent and a B from the other, your blood isn't some halfway type. Nope, you have AB blood type. Both the A and B markers are fully expressed on your cells. So incomplete is a blend, codominance is both. So for a long time after Mendel, there was still this huge question hanging in the air. What are these factors he talked about? Where are they hiding in the cell? Mendel knew they existed, but he couldn't point to them. He was missing the physical proof.
Then, in the early 1900s, scientists like Walter Sutton and Theodore Boveri are looking at cells dividing under the microscope. And they notice something that makes the hairs on their arms stand up. The chromosomes they're watching, they behave in the exact same way as Mendel's imaginary factors. They come in pairs, they separate when sperm and eggs are made, different pairs sort on their own. It was a perfect one-to-one match.
And this was it. This was the aha moment that led to the chromosomal theory of inheritance. It was the grand unification theory of genetics, connecting Mendel's abstract laws with the physical things, the chromosomes, you could actually see in a cell. Finally, the mystery of where was solved.
Okay, peas, flowers, chromosomes, that's all great. But what about us? Let's bring this story home and see how these exact same rules play out in you and me.
A really clear example is how sex is determined in humans. Females have 2 X chromosomes. Males have 1 X and 1 Y. A mother can only pass on an X, so it's all down to the sperm. If it carries an X, the baby is female. If it carries a Y, the baby is male. Simple as that. And every single time, it's a coin flip, a 50-50 chance.
And this is where Mendel's work goes from being about garden peas to saving human lives. These predictable patterns are exactly how we understand many inherited genetic disorders. Conditions like hemophilia, which is a bleeding disorder, or sickle cell anemia. They're called Mendelian disorders for a reason. It's because a mutation in just one single gene follows these exact rules, which lets us track them through families and understand the risks.
So we started with a simple question about elephants and ended up decoding the blueprint for all life. This incredible instruction manual passed down through countless generations. Which leaves us with one last, massive question to chew on. As we get better and better at not just reading this blueprint, but maybe even rewriting it, what does that future look like?